In this module, we will be covering types of sequence variation in the human genome. The expected learning outcomes for this module are to learn about types of sequence variation present in the human genome, and discover how these genetic changes can impact specific traits and phenotypes. Now that the Human Genome Project was completed, our attention shifted to understanding sequence variation, or in other words, sequence differences between individuals. There are a number of projects that were implemented to get this type of work done. These include the Human HapMap project, which ran from 2002 to 2009. Now, the Human HapMap project aimed to describe the common patterns of variation across the human genome by looking at a specific type of variation known as a SNP, which we'll learn about in the next slide. And more recently, the 1000 Genomes project that started in 2008 to provide comprehensive human genetic variation resources by sequencing over 1,000 individuals. Now, you may be asking, when we did all of these comparisons among individuals, what did we find? The answer is we found a number of different types of sequence variation. One of the most common types of sequence variation that we identified was single nucleotide variants. Now, these are simply sites in the DNA sequence where individuals differ at a single DNA base. Very closely related to the single nucleotide variant is the single nucleotide polymorphism, or SNP. Now, these are single DNA base changes, but at a specific frequency in the population, generally over 1%. And SNPs are the most common form of genetic variation. And you see a nice example of a SNP over to the right, where individual 1 has an A allele. Remember that an allele is simply a specific version of a SNP or gene. So individual 1 has an A allele at this specific position in the genome. Individual 2 has a G allele, and individual 3 has a T allele. SNPs that fall within genes may lead to some fairly drastic changes in phenotype. There are several different types of point mutations. These include things like synonymous mutations, or silent, that actually do not result in a change to the amino acid sequence. You can see a nice example of a silent mutation here, where we've changed the original C allele to a T allele, but when it's transcribed into mRNA and translated into protein, it still is translated as this lysine amino acid. There are also times when SNPs cause non-synonymous mutations. These can include things like missense mutations, where changes in the DNA base result in a change in the amino acid. You can see a good example of two missense mutations here, where we've changed the T allele to a C that causes an arginine residue to be translated, and changing the T allele to a G, causing a threonine amino acid to be translated. And the last type is known as nonsense mutation, which results in the creation of a premature stop in the protein. And you can see a good example in the middle pane here, where we've changed the T allele to an A allele, causing the stop signal to the translation of the protein. Now, a good example of a missense mutation is in sickle cell anemia, where changing this T allele to an A allele at a particular position in the hemoglobin gene is actually the cause of sickle cell disease. And you can see that it changes this glutamic acid residue to a valine residue, causing the mutant phenotype of sickling in the red blood cells. Another type of sequence variation that was seen is called structural variation. Now, structural variation relates to the size of the genetic variant. It can be defined as genetic variation that occurs over a large DNA sequence. This is generally about 1,000 base pairs or more. And you can see some common types of structural variation in the figure to the right. Now, a common example of structural variation and how it can impact specific phenotypes and traits is that large structural variations can often lead to specific types of cancer.